Hello students, today we are going to discuss in detail on genome mapping, in which how the various techniques such as FISH, RFLP or APD are involved. First of all, we'd like to know what is a genome. Genome is an applied set of chromosomes in a gamete or microorganism or in each cell of the multicellular organism. It is encoded either DNA or in either DNA, that is the case of the eukaryotes, and the case of the prokaryotes, with the exception of viruses, DNA is present as the genetic material, whereas in the case of the viruses, some of them are having RNA as genetic material. We call them as retroviruses. Each genome contains all of these information needed to build and to maintain the organism. Each genome contains all of the information that is needed to build and maintain the organism. We know very well the case of the nucleic DNA. In overall total chromosome, we have 3.2 into 10 to the power 6, 10 to the power 9 billion base pairs are present in the case of our system. It's very difficult task to create a map of every gene which is scattered over the all 46 chromosomes. A graphic what is genome mapping? A graphic representation of the arrangement of gene on DNA sequence on the chromosome. So the human genome map was completed in 1996, locates nearly 5264 markers for a particular gene. Normally, a genome map is used to identify and to record the location of the gene and the distance between the gene of the chromosome. There are various types of genome mapping is available. Normally, we come across two types of genome maps are known. Genetic or linkage maps. Another one is physical maps. Both the maps are collection of genetic markers. We know very well what are genetic markers that are associated with the chromosome. It is short-term repeats and long-term repeats, as well as interspersed repeats. It is NTR, VNTR, STR, as well as sign, line, and allo elements. All these are called as genetic markers for a particular genome. First of all, we'd like to know what is meant by linkage or genetic mapping. The genetic map illustrates the order of the gene on a chromosome and the relative distance between the one gene to the other gene. So, just like finding a distance between one place to the other place where it is situated. So, genetic maps are based on recombination, that is, exchange of DNA sequences between non-sister chromatids during the process of a meiosis is considered as the genetic map under linkage and recombination process. A genetic map is prepared on the basis of recombination of data between carefully selected genetic markers, usually ordered into suitable crosses. Here is the recombination frequency, the number of recombinant progeny into 100 by total number of progeny, that is the number of recombinant genes which is present divided by the total number of progeny we consider. But in the case of the human being, a linkage map has to be prepared using a family pedigree, how the particular gene is inherited from one generation to the another generation. For example, hemophilia and color blindness. Hemophilia is a clinical condition in which loss of blood takes place because due to lack of clotting factor. In the same manner, in the case of the color blindness, they cannot differentiate between the colors, one of the colors, during the reading in the eye. So, where the color blindness will be there in the case of the person that is inherited from one generation to the next generation. Here we can see a pedigree, how it is one of the parent is having, that is a father is having the genetic disorder, how it is inherited to the first generation as well as in the second generation, how many of them are getting the clinical condition. In such map, the distance between the genes are shown in the case of map units, or we call it as centimorgan, because it was identified by the Morgan. So it is named after the scientist centimorgan. The chief problem of linkage mapping is non-availability of sufficient number of the genetic markers to cover the entire genome. That is a problem. This was solved by the physical mapping. The physical mapping provides the detail of the actual physical distance between the genetic marker as well as the exact location of the gene. That is done by a physical mapping. There are three most important techniques that are used to create a physical map: somatic cell hybridization, radiation hybridization, fluorescent in situ hybridization. Somatic cell hybridization is also called as somatic cell fusion or protoplast fusion. The development of hybrid plants through the fusion of somatic protoplast of two different plant species is called as somatic hybridization. That is, body cells are utilized, not the germline cells are utilized for this purpose. So we call it as somatic cell fusion. And protoplast means the cell wall is eliminated and the cell divide of cell wall in a plant is in turn fused together. We call it as somatic hybridization. That is involving following steps: isolation of the protoplast, fusion of the protoplast of the two different species, and selection of the somatic hybrids, then culturing them, and regeneration of the hybrid plants from them. This is the technique followed in the case of the plant. But in the case of the animal cell, we take up human fibroblast and mouse cell tumor cells, and they are fused together. Since it is a totally different species, mouse is the animal species and human is different. When they fuse together, we are getting the nucleus of the two cells mixed up together to form the heterokaryote. Then these heterokaryotes are then placed in the medium of HAT, that is hypoxanthine and thymine medium, in which proliferation of the hybrids forms the colony on the selective medium. So this is the hybrid, where we come across the hybrid colony of A, B as well as C in this case, from which we can select the cells and to identify. The next is the method. What we come across is nothing but the method of high resolution mapping, called as a radiation hybridization, in which the radiation hybrid it involves the usage of your X-ray that breaks the DNA into fragments. The amount of radiation can be adjusted to create either small fragments of DNA or large fragment of DNA. This technique is not affected by or increased or decreased of recombination frequency. The next one is your fluorescent in situ hybridization. In short, we call it as FISH technique. It's a powerful technique for detecting DNA RNA sequence in the case of cells, tissues, and tumors. FISH provides a unique link among the studies of cell biology, cytogenetics, as well as molecular genetics. This is providing spatial resolution of morphological and genomic structure. This is a rapid technique and important, and offers great probe stability. That is, in this method we are using fluorescent probes for this purpose. They are different types of probes are utilized. They are locus specific probes or intercalating probes are utilized. Normally the locus specific probes, they bind to the particular region of the chromosome. This type of probe is useful for the researcher to isolate a small portion of the gene and to determine on which gene the chromosome is located. For example, alphoid or centromeric repeat probes are generally used for repetitive sequence that is found in the middle of the chromosome, that is in the centromeric region, whereas whole chromosome probes are actually a collection of small probes which binds at different sequences along the entire length of the DNA. It can bind with the entire length of the DNA. We call it as whole chromosome probes. Fluorescent in situ hybridization, the DNA is cut and the fluorescent probes are added with the help of your DNA polymerase bionic translation technique. Then it is denatured by 75 degrees centigrade. Then hybridization is done by infusing the cells, by denaturating and forming. Then later, antibodies or anti-Dick or abidin is linked with the fluorescent probe that in turn creates a fluorescent structure of the particular uh, cell in this case, fixed cells in this case. This is how we can create the fluorescent in situ appreciation technique in the case of a particular chromosome where we can detect it. What is application? Fish has large number of applications in molecular biology and medical science. It is useful in gene mapping, diagnosis of chromosomal abnormalities, and to study of cellular structure and function. In the clinical research, it is especially used in prenatal diagnosis and cancer diagnosis, and it also useful in identifying molecular cytogenetic birth defects and mental retardation in the case of the children, and diagnosis of infectious diseases and detection of aberrant gene expression. All these are made possible by our fish technique. In the laboratory research, the fish can be used to study the evolution of genome and to analyze nuclear organization, visualization of chromosomal territories and chromatin, etc. Even it is useful in creating chromosome painting, where we make our chromosome colorful by using the fluorescent markers. So these molecular markers are fragments of DNA that is associated with certain locations in the genome that we can able to identify. How to identify these molecular markers? In case of the DNA, already we know very well. First and foremost technique of molecular marker is nothing but RFLP. RFLP is a genetic marker, that is restriction fragment length polymorphism. It's a genetic marker that can be examined by cleaving the DNA into fragments with the help of our restriction enzyme. The restriction enzyme cuts the DNA at a particular sequence. For example, in 3, that cuts at the sequence of GTCCAA, that is a sequence, that is palindromic sequence. If we apply the restriction enzyme on DNA, it cuts at every occurrence of this particular palindromic sequence into million restriction fragments to a few thousand nucleotide longs. Then once the DNA is cut by this technique, then later the DNA fragments are taken up and it is added with the radioactive probes that binds with the specific DNA fragment. Then it is transferred to the DNA southern blotting techniques. When it is electrophoresed, the smaller fragments move faster when compared to the larger fragments, and the DNA pattern is created that is in turn compared with the known subjects after applying nitrocellulose membrane. This pattern get transferred to nitrocellulose membrane that creates that it is placed over the X-ray film that is sandwiched between the nitrocellulose membrane for the to identification. This DNA pattern is compared with that of the known subjects. From that we can able to identify what is the repetitive sequence found in the case of this, or the marker found in this. RFLP is mainly as lot of application. It is useful in identification of and isolation of any known gene, any gene known to be linked with the RFLP locus. It is normally useful in identifying paternity cases and criminal cases to identify the culprit as well as the identification of the lost child from the parents. And it is also useful in identifying loci affecting quantitative tract, and further it is useful in determination of chromosomal segments, which yields the best result. Diagnostic genetics, it can be helpful in identifying genetically inherited disorder diagnosis. It is useful in identifying hybrids, pedigree analysis, and genetic screening. So this is the advantages of our RFLP. And there are some disadvantages. It is highly expensive. It requires relatively large amount of DNA. but low level of polymorphism in some species. It needs a suitable probe library and time consuming. RAPD. RAPD is random amplification of polymorphic DNA. It is a PCR-based technology that is developed by Welsh and McClellan. This procedure detects nucleotide sequence polymorphism in DNA. It detects the dominant variation in the genome. It is used to analyze the genetic diversity of an individual by random primers. H1, H2 genome, and a primer is added, the DNA is amplified. Then solution 1 and 2, when it is transferred to the agarose gel, where we can get the fragments. It is useful in gene mapping, DNA amplification, fingerprinting, and to study close related species can be analyzed with the help of our RAPD. They are quick and easy to assay because PCR is involved. Only low quantity of DNA is more than sufficient to carry out this mechanism. And RAPD has a very high genomic abundance and are randomly distributed throughout the genome. Pure DNA is not at all needed. Disadvantage: the markers are dominant. They cannot be distinguished whether the DNA sequence is amplified from the locus, that is homozygous or heterozygous. Since it is involving PCR enzymatic reaction, the components are greatly influencing the outcome. Some mismatches between the primer and template takes place during this process and results in total absence of PCR product. And moreover, the RAPD results are difficult to interpret. These are all the application of our RAPD. So here some basic difference between the RFLP and as well as RAPD. And this is our references. Thank you.